Hello, my wonderful and lovely humans. So today we are going to be making an enzyme foldable. So you guys should have this piece of paper. And if you look at both sides, there are lines here, and it doesn't really matter which way you fold it, but try to fold it in on the lines. But basically, once you have that folded down, what you're supposed to have is a little tri-fold here. And the main idea behind this is so that you can see the two pictures. Oh, sorry, 4 pictures on the outside, okay? Because enzymes are a 4-step process, all right? Okay, so make sure you have your page set up like this. You are also gonna need about 5 different colors. Hopefully I would have already told you that. But you guys are gonna need about 5 different colors. And yes, I do want you guys to color because it's just gonna help you to keep things straight, okay? All right, so let me go ahead and switch my camera. Okay, awesome. So hopefully your page looks like this. If it doesn't, make sure it does, okay? Don't worry, you guys know that I will always, always, always show you the finished product at the end of the class, okay? All right, so let's talk about this 4-step process. So our first step is what we call the lock and key step, okay? And that is because it introduces us to that idea that the enzyme has a lock and key function. So up here, what we have is called a substrate. This thing right here is a substrate. All right. And a substrate will fit into what we call an enzyme down here. I know I wrote my Z weird. Sorry, I couldn't help myself. But anyways, so the lock and the key, basically the substrate will fit into the enzyme. So the substrate will act as though it's a key, and the enzyme will act as though it's a lock. So our substrate will fit into our lock. And I'm going to make sure that I also update the colors of these guys. And you know what? Since I'm doing my enzyme right now, I'm going to go ahead and skip ahead and do all of my enzymes, because guess what? That enzyme shows up in every single picture. So just skip ahead really quickly and make sure that you are filling in your enzyme. There we go. Okay. All right, a little sloppier than I would personally like, but you know, whatever. Okay, so like I said, the substrate's gonna fit into the enzyme. Now just like a key and a lock, if that key does not fit the lock correctly, guess what? That thing is not gonna get unlocked, okay? So that substrate is very specific to that enzyme. All right, so the next step is what we call the enzyme substrate complex. Substrate complex. And that is step number 2 over here, okay? So the enzyme substrate complex is where the enzyme actually does fit into our, or sorry, our substrate is going to fit into our enzyme, and it's going to meet at this place that is called an active site. The active site is where the action is happening. That's why it's active, right? So in green over here, oops, and I did not grab my green pen. Let me grab it. Okay. But in green over here, I've marked the active site. And if you guys notice what's happening over here, we are having like a breakdown of this original substrate. And this original substrate is now becoming 2 different molecules. So I'm gonna go ahead and make sure that I am including those new, a new color so that it basically kind of emphasizes the idea that we have new products. And that should have probably been this one. Awkward. All right, but as you guys can see, I have two new colors. If you look down here, this specific image is going to label it as 1 and 2. So I'm going to go ahead and label these also as 1 and 2, which means that the colors that I'm going to fill in over here are going to match up with those guys up in number 2. So I'm going to just skip ahead and fill it in. Oops, almost did the wrong color again. There we go, teal. Sorry guys, multiple colors can be difficult to keep track of, I guess. All right, and I just wanted to make sure that you guys can see the 1 and the 2. But yeah, so what we end up with down here is what we know as a product. Okay, we end up getting a product, and that product is brand spanking new. It has not existed up until this point. However, if you guys notice, remember, we're still keeping that same enzyme. So the enzyme remains the same. Enzyme remains the same. All right. And what we're going to call this step is the enzyme and products. This is our enzyme and product stage. So we still have the active site over here. However, this enzyme has already done its job, so it's not necessarily ready to accept another one quite just yet. Well, that is until we get to step number 4. And in step number 4, well, guess what? Our enzyme is basically ready to start back at step 1 and is ready to do it all over again. All right. So our enzyme will return back to its state, and it's going to be ready to do it all over again. Go through this process, starting back at step 1 and moving through steps 2, 3, and well, 4 is basically step 1, right? But that means that this substrate that's up here is the same substrate that was back up there. So I'm going to return to that original color. Okay. So if you guys need to, go ahead and pause the video so you guys can get this copied down. We are going to next move on to the inside of this foldable, and you guys have a few more notes to go, so just stick with me and we will make it through. But if you need to, go ahead and pause the video. Okay, so hopefully you guys have had enough time. If not, well, you know, figure it out. Basically, I don't know. I might have better advice for you in the future. But let's go ahead and move on over here to the inside of our little foldable. Okay, so we have what is known as the lock and key model here. So what is happening here is that, okay, and I'm going to try to keep up my colors. You guys don't have to. It's just, you know, I think that it's pretty. But I used orange for substrate. So I'm going to use orange for substrate again. So substrates and enzymes. And because I use pink for enzymes, I'm going to use pink again. Substrates and enzymes have complementary shapes. Okay, and those complementary shapes we should have covered in our original vocab, but complementary is going to mean that they fit together. They were made for each other, right? So complementary shapes. Let's do blue. Complementary shape means that they fit together and were made for each other. All right. Substrates and enzymes have complementary shapes. That means that they fit together and basically that they were made for each other. Okay. All right. So the other thing is that substrates and enzymes fit together. So let me do my coloring again. Sorry, trying to keep track of the colors. Okay, substrates and whoops, I'm just throwing things around. And enzymes are going to fit together like a lock and key. So do you guys get the idea that this lock and key model is pretty important to us? So basically that means if the enzyme, or sorry, if the substrate is the wrong substrate, it will not work. If substrates is the wrong one, the enzyme will not work. Sorry, I ran out of space there. But the enzyme will not work if the substrate is the wrong one, okay? All right. And lastly, the last important detail here is that each enzyme will only catalyzes one substrate. Okay, that means that each enzyme is going to only have one substrate that was made for it. So for basically each lock, there is only one key. All right, and I'm going to zoom in on this so you guys can pause the video and copy it down. All right, so if you need to, go ahead and pause the video. Okay, we got a little bit more writing to do, guys, but we are getting closer and closer, so stick with me. We are almost there, okay? All right, so we need to talk about a few characteristics of enzymes because enzymes are important. I told you guys this. Enzymes are super important for us just to survive, to continue living, and basically they're going to make reactions happen without having to use so much energy. So that is really important for us. So the first characteristic you guys need to know is that enzymes can be reused. All right, enzymes can be reused. So it is really important to know that enzymes will not get consumed by this process. They are not used up. In fact, they end up existing at the end, which is why we were able to return back to step one. So they are constantly being reused. Okay, number two is that enzymes are specific. Okay, they are specific, meaning that back to this idea over here, for each lock, there is only one key. So a different fitting key will not fit into that lock. It will not do the function that it needs to do. Okay. That's number 2. Number 3. Enzymes work by lowering the, I don't know what color to do, so I'll do this one in green. They work by lowering the activation energy Okay, they work by lowering the activation energy that is required. So if you guys remember that speed bump from the image that I made you guys draw, so I believe, wait a second, I'm gonna have to pull out my notebook here. So I guess that gives you guys a second to copy if you need to. But I do want to show you that graph that I had you guys copy from your notes. So here it is. But as you guys remember, so the activation energy, so you guys see 2 different activation energy requirements here. This black line is going to basically show you how much activation energy would be required if we did not have an enzyme. But this little blue line shows us that if we do have an enzyme, it requires a lot less activation energy. That just means that we have to use up less of our So that is what enzymes are going to do. They work by lowering the activation energy requirement. All right, number 4. Okay, so enzymes are going to make materials that cells require. Enzymes will make materials that cells require. So your cells, your body cannot function without enzymes because they literally won't have the materials to exist, to sustain themselves, and to work. So enzymes will make those materials, and another important reason why we have enzymes. And lastly, we have number 5. And number 5 is enzymes. Enzymes assist in breaking down molecules and bonds. And why do I write bonds in green? Because when we break bonds, we release what? Yes, you need to answer. You release what, guys? You release energy. And I'm going to write that nice and big. Okay? So enzymes are going to assist in breaking down molecules and bonds. And when we break those bonds, we release energy. All right. So if you guys need to, go ahead and pause the video so you guys can copy this down. We are going to be finishing up this last section here. And guys, stay with me. It is not that much worse from here on out. We are super close. Okay. All right, so now we should be ready to move on to what affects how enzymes work. So let's talk about it. So there are 3 different things that can affect it. So 1, 2, 3. Okay. All right, so the first thing that can affect how enzymes work is going to be temperature. Okay, so basically if the enzyme becomes too hot, comma, it will denature. All right, denature, and denature means to fall apart. Okay, so if the enzyme becomes too hot, it will denature or fall apart. The second thing that can affect how an enzyme will work is pH level. So pH is going to refer to the acidity or the basicness. So acidity meaning if you guys were to like lick a lemon, right, you would taste really like acid. But if you, I don't know, eat dark chocolate, on the other hand, that might be kind of bitter to you. But anyways, pH can definitely affect our enzymes. It can be too acidic. And once again, we can experience that denaturing. Can be too acidic. Okay. And the third and final thing that will affect how an enzyme works is what we call the inhibitor. And the inhibitor is actually going to be something that we do like because this is going to act as our brake. So the inhibitor is the brakes that will stop a reaction. Okay, and don't forget that RXN means reaction. Okay, oops. All right, so temperature, pH, and inhibitors. Okay, these three things will affect how enzymes work. Inhibitor, the last one specifically, is something that we're going to use in our bodies to specifically stop this process. Okay. All right, so that is all I have for you guys today. I guess let me zoom out here. So if Miss Reyes needs to, she can pause the video, I guess, and you guys can get a chance to copy this down. All right, but yep, that's all I have for you guys today. Okay, take it over, Miss Reyes. Bye.